Class 12, Chapter 4 Principles of Inheritance and Variation. Have you ever wondered why an elephant always gives birth only to a baby elephant and not some other animals, or why a mango seed forms only a mango plant and not any other plant? Given that they do, are the offspring identical to their parents, or do they show some difference in some of their characteristics? Have you ever wondered why siblings sometimes look so similar to each other or sometimes even so different? These and several related questions are dealt with scientifically in a branch of biology known as genetics. This subject deals with the inheritance as well as the variation of character from parents to offspring. Inheritance is the process by which characters are passed on from parents to progeny. It is the basis of heredity. Variation is the degree by which progeny differ from their parents. Humans knew from as early as 8000 to 1000 BC that one of the cause of variation was hidden in sexual reproduction. They exploited the variation that were naturally present in the wild population of plants and animals to selectively breed and select for organisms that possess desirable character, for example through artificial selection and domestication from ancestor wild cows. We have well-known Indian breeds, example Sahiwal cows in Punjab. We must, however, recognize that though our ancestors knew about the inheritance of characters and variation, they had very little idea about the scientific basis of their phenomena. 4.1 Mendel's law of inheritance. It was during the mid 19th century that headway was made in the understanding of inheritance. Gregor Mendel conducted hybridization experiment on garden pea for 7 years 1856 to 1863 and proposed the laws of inheritance in living organisms. During Mendel's investigation into inheritance pattern, it was for the first time that statistical analyzing and mathematical logics were applied to problems in biology. His experiment had a large sampling size which gave greater credibility to the data that he collected also. The confirmation of his inference from experiment on successive generation of the test plants proved that his results pointed to general rules of inheritance rather than being unsubstantiated ideas. Mendel investigated characters in the garden pea plant that were manifested as two opposite characters, example tall or dwarf plant, yellow or green seed. This allowed him to set up a basis framework of rules governing inheritance, which was expanded on by later scientists to account for all the diverse natural observations and the complexity inherent in them. Mendel conducted such artificial pollination cross pollination experiment using several true breeding pea plant lines. A true breeding line is one that, having undergone continuous self pollination, shows the stable trait inheritance and expression for several generations. Mendel selected 14 true breeding pea plant varieties as pair which were similar except for one character with contrasting traits. Some of the contrasting traits selected were smooth or wrinkled seed, yellow or green seed, inflated full or constructed green or yellow pods, and tall or dwarf plant. Figure 4.1, Table 4.1. Figure 4.1, 7 pairs of contrasting trait in pea plants studied by Mendel. Table 4.1, Contrasting traits studied by Mendel in pea. 4.2, Inheritance of one gene. Let us take the example of one such hybridization experiment carried out by Mendel, where he crossed tall and dwarf pea plant to study the inheritance of one gene. Figure 4.2. He collected the seeds produced as a result of this cross and grew them to generate plants of the first hybrid generation. This generation is also called the filial progeny or the F1. Mendel observed that all the F1 progeny plant were tall like one of its parents, none were dwarf. Figure 4.3. He made similar observation for the other pairs of trait. He found that the F1 always resembled either one of the parent and that the trait of the other parent were not seen in them. Mendel then self-pollinated the tall F1 plant and to his surprise found in the filial second generation some of the offsprings were dwarf. The character that was not seen in the F1 generation was now expressed. The proportion of plant that were dwarf were 1 by 4th of F2 plant while 3 by 4th of F2 plant were tall. The tall and dwarf trait were identical to their parental type and did not show any blending, that is, all the offspring were either tall or dwarf. None were in between height. Figure 4.3. Similar results were obtained with the other trait that he studied. Only one of the parental trait was expressed in the F1 generation, while at F2 stage both the traits were expressed in the proportion 3:1. The contrasting trait did not show any blending at either F1 or F2 stage. Figure 4.2. Steps in making a cross in P. Based on these observations, Mendel proposed that something was being stably passed down unchanged from parent to offspring through the gamete over successive generation. He called these things as factor. Now we call them as genes. Genes therefore are the unit of inheritance. They contain the information that is required to express a particular trait in an organism. Genes which code for a pair of contrasting traits are known as allele. They are slightly different from of the same gene. If we use alphabetical symbol for each gene, then the capital letter is used for the trait expressed at the F1 stage and the small alphabet for the other trait. For example, in case of the character of height, capital T is used for tall trait and small t for the dwarf, and capital T and small t are alleles of each other. Hence, in plant, the pair of alleles for height would be capital T, small t, capital T, or small t. Mendel also proposed that in a true breeding, tall or dwarf P variety, the allergic pair of genes for height are identical or homozygous, capital T, and small t, respectively. Capital T and small t are called genotype of the plant, while the descriptive terms tall and dwarf are the phenotype. What then would be the phenotype of a plant that had a genotype capital T small t? As Mendel found the phenotype of the F1 heterozygote capital T small t to be exactly like the capital T parent in appearance, he proposed that in a pair of dissimilar factor, one dominates the other as in the F1 and hence is called the dominant factor, while the other factor is recessive. In this case, capital T for tallness is dominant over small t for dwarfness, that is recessive. He observed identical behavior for all the other character trait pairs that he studied. It is convenient and logical to use the capital and lower case of an alphabetical symbol to remember this concept of dominance and recessiveness. Do not use capital T for tall and D for dwarf, because you will find it difficult to remember whether capital T or small d are alleles of the same gene character or not. Alleles can be similar as in the case of homozygotic capital T and small t, or can be dissimilar as in the case of heterozygote capital T small t. Since the capital T small t plant is heterozygous for genes controlling one character height, it is a monohybrid, and the cross between capital T and small t is a monohybrid cross. From the observation that the recessive parental trait is expressed without any blending in the F2 generation, we can infer that when the tall and dwarf plant produce gametes by the process of meiosis, the alleles of the parental pair separate and segregate from each other, and only one allele is transmitted to a gamete. This segregation of allele is a random process, and so there is a 50% of chance of a gamete containing either allele, as has been verified by the result of crossing. In this way, the gametes of the tall capital T plant have the alleles capital T, and the gametes of the dwarf small t plant have the alleles capital T. During fertilization, the two alleles capital T from one parent, say through the pollen, and small t from the other parent, then through the egg, are united to produce zygote that have a capital T allele and one small t allele. In other words, the hybrid have small t and capital T. Since these hybrid contain allele which express contrasting traits, the plants are heterozygous. The production of gametes by the parents, the formation of the zygote, the F1 and F2 plant can be understood from a diagram called Punnett square. As shown in figure 4.4, it developed by a British scientist, geneticist Reginald C. Punnett. It is a graphical representation to calculate the probability of all possible genotype of offsprings in a genetic cross. The possible gametes are written on two sides, usually the top row and left column. All possible combinations are represented in boxes below in the square, which generate a square output form. The Punnett square shows the parental tall capital T male and dwarfs small t female plant, the gametes produced by them, and then F1 capital T small t progeny. The F1 plant of genotype capital T small t are self-pollinated. The symbols male and female are used to denote the female eggs and male pollen of the F1 generation respectively. The F1 plant of the genotype, when a typical monohybrid self-pollinated produce gamete of the genotype capital T and small t in cross conducted by Mendel proportion. When fertilization takes place, the pollen grain of genotype true breeding tall plant capital T have a 50% chance to pollinate eggs of the genotype capital T as well as of genotype small t. Also, pollen grain of genotype small t have a 50% of chance of pollinating eggs of genotype capital T as well as genotype small t. As a result of random fertilization, the resultant zygote can be of the genotypes capital T small t or small t. From the Punnett square, it is easily seen that 1/4 of the random fertilization rate to capital T 1/2 lead to capital T small t and 1/4 to small t. Through the F1 have a genotype of capital T small t, but the phenotype character seen in is tall. At F2, 3/4 of the plant are tall, where some of them are small t capital T while other are capital T small t. Externally, it is not possible to distinguish between the plant with the genotype capital T and capital T small t. Hence, within the genotypic pair capital T small t, only one character capital T tall is expressed. Hence, the character capital T or tall is said to be dominant over the other allele small t or dwarf character. It is just due to the dominance of one character over the other that all the F1 are tall through the genotype is capital T small t, and in the F2, 3/4 of the plant are tall through genotypically 1/2 are capital T small t and only 1/4 are capital T. This lead to a phenotypic ratio of 3/4 tall, 1/4 capital T plus 1/2 capital T small t and 1/4 small t. A 3:1, but a genotypic ratio of 1:2:1. That 1/4 ratio, 1/2 ratio, 1/4 ratio of the capital T small t ratio small t is mathematically condensable to the form of a binomial expression ax plus by whole square that has a gamete pairing genes capital T or small t in equal frequency of 1/2. The expression is expanded as given below. 1/2 capital T plus 1/2 small t का whole square is equals to 1/2 capital T plus 1/2 small t into 1/2 capital T plus 1/2 small t is equals to 1/4 capital T plus 1/2 capital T small t plus 1/2 small t. Mendel self-pollinated the F2 plant and found that dwarf F2 plant continued to generate dwarf plant in F3 and F4 generation. He concluded that the genotype of the dwarf was homozygous small t. What do you think he would have got had he self-pollinated a tall F2 plant? From the preceding paragraph, it is clear that through the genotype ratios can be calculated using mathematical probability. By simply knowing at the phenotype of a dominant trait, it is not possible to know the genotype composition. That is, for example, whether a tall plant from F1 or F2 has capital T or small t capital T. Composition cannot be predicted. Therefore, to determine the genotype of a tall plant at F2, Mendel crossed the tall plant from F2 with a dwarf plant that he called a test cross. On a typical test cross, an organism P plant here showing a dominant phenotype and whose genotype is to be determined is crossed with the recessive parent. Instead of self-crossing, the progenies of such a cross can easily be analyzed to predict a genotype of the test organism. Figure 4.5 shows the result of the typical test cross where violet color flower capital W is dominant over the white color flower color small w. Using Punnett square, try to find out the nature of the offspring of a test cross. What do you ratio got? Using the genotype of this cross, can you give a general definition for a test cross? Based on his observation on monohybrid cross, Mendel proposed two general rules to consolidate his understanding of inheritance in monohybrid cross. Today, these rules are called the principle of laws of inheritance. The first law of or law of dominance, and the second law or law of segregation. 4.2.1 Law of dominance. First, characters are controlled by discrete unit called factor. Second, factor occurs in pair. In a dissimilar pair of factor, one member of the pair dominates the other recessive. The law of dominance is used to explain the expression of only one of the parental character in a monohybrid cross in the F1 and the expression of both in the F2. It also explains the proportion of 3:1 obtained at the F2. 4.2.2 Law of segregation. This law is based on the fact that the alleles do not show any blending and that both the characters are recovered as such in the F2 generation. Though one of these is not seen at the F1 stage, though the parent contains two alleles during gamete formation, the factor or alleles of a pair segregate from each other such that a gamete recessive receive only one of the two factors. Of course, a homozygous parent produce all gametes that are similar, while a heterozygous one produce two kinds of gametes, each having one allele with equal proportion. 4.2.2.1 Incomplete dominance. When experiments on peas were repeated using other traits in other plants, it was found that sometimes the F1 had a phenotypic that did not resemble either of the two parents and was in between the two. The inheritance of flower color in the dog flower snapdragon or Antianum sp. is a good example to understand incomplete dominance. In a cross between true breeding, red flower R are true between true breeding. White flower plant smaller. The F1 R smaller was pink. Figure 4.6. When the F1 was self-pollinated, the F2 resulted in the following ratios: 1 capital D capital R red ratio 2 capital R pink ratio 1 smaller white. Here the genotype ratios were exactly as we could expect in any Mendelian monohybrid cross, but the phenotypes ratio had a change from 3:1 dominant recessive ratio. What happened was that capital R was not completely dominant over small r, and this made it possible to distinguish capital R smaller as pink from smaller and capital R. Explanation of the concept of dominance. What exactly is dominant? Why are some alleles dominant and some recessive? To tackle these questions, we must understand what a gene does. Every gene, as you know by now, contains the information to express a particular trait. In a diploid organism, there are two copies of each gene as a pair of alleles. Now, these two alleles need not only to be identical, as in heterozygous, one of them may be different due to some changes that it has undergone, about which you will read that it has undergone further on, and in the next chapter you will modify the information that particular allele contain. Let's take an example of the gene that contain the information for producing an enzyme. Now there are two copies of this gene, the two allelic forms. Let us assume, as in more common, that the normal allele produce the normal enzyme that is needed for the transformation of the modified allele could be responsible for the production of normal or less efficient enzyme or a non-functional enzyme or no enzyme at all. In the first case, the modified allele is equivalent to the unmodified allele. It will be produce the same phenotype trait, result in the transformation of substrate. As such, equivalent allele pair are very common, but if the allele produce a non-functional enzyme or no enzyme, the phenotype may be affected. The phenotype trait will only be dependent on the functioning of the unmodified allele. The unmodified functional allele, which represent the original phenotype, is the dominant allele, and the modified allele is generally the recessive allele. Hence, in the example above, the recessive trait is seen due to non-functional enzyme or because no enzyme is produced. 4.2.2.2 Codominance. Till now we were discussing crosses where the F1 resembled either of the two parent dominance or was in between incomplete dominance, but in the case of codominance, the F1 generation resemble both parents. A good example is different type of red blood cell that determine ABO blood grouping in human beings. ABO blood grouping are controlled by the gene I. The plasma membrane of the red blood cell has sugar polymerase that produced from its surface, and the kind of sugar is controlled by the gene. That gene I has three alleles IA, IB, and small I. The alleles IA and IB produce a slightly different form of the sugar, while allele small I does not produce any sugar. Because humans are diploid organisms, each person possesses small I. In other words, when capital IA and small I are present, only IA expressed because I does not have any sugar, and when IB and small I are present, IB express. But when IA and IB are present together, they both express their own types of sugar. This is because of codominance. They hence red blood cell have both A and B type of sugar. Since there are three different types of alleles, there are six different combination of these three alleles that are possible, and therefore a total of six different genotypes of the human ABO blood types. Table 4.2. How many phenotypes are possible? Do you realize that the example of ABO blood grouping also provide a good example of multiple allele? Here you can see that there are more than two allele, three alleles governing the same character. Hence, in an individual, only two alleles can be present. Multiple alleles can be found only when population studied are made. Occasionally, a single gene product may produce more than one effect. For example, starch synthesis in a pea seed is controlled by one gene. It has two alleles, capital T and small b. Starch is synthesized effectively by capital B homozygous, and therefore large starch grain are produced. In contrast, small b homozygote have lesser efficiency in starch synthesis and produce smaller starch grain after maturation of the seed. Small B seeds are round, and the small b seeds are wrinkled. Heterozygous produce round seeds, and so capital B seems to be dominant allele, but the starch grain produced are of the intermediate size in capital B seeds. So if starch grain size is considered to be the phenotype, then from the angle the alleles show incomplete dominance. Therefore, the dominance is not an autonomous feature of a gene or the product that it has information for. It depends as much on the gene product and the production of a particular gene phenotype from this product as it does not on particular phenotype that we'll choose to examine in case more than one phenotype is influenced by the same gene. 4.3. Inheritance of two genes. Mendel also worked with and crossed pea plant that different in two character as in seen in the cross between a pea plant that has seed with yellow color and round shape, and one that had seeds of green color and wrinkled shape. Figure 4.7. Mendel found that the seeds resulting from the crossing of the parent had yellow colored and round shaped seeds. Here, can you tell which of the character in the pair's yellow, green color and round wrinkled shape was dominant? Thus, yellow color was dominant over green and round shape dominant over wrinkled. These results were identical to those that he got when he made separate monohybrid cross between yellow and green seeded plant and between round and wrinkled seeded plant. Let us use the genotype symbol capital Y for dominant yellow seed color and small y for recessive green seed color. Capital R for round shaped seed and small r for wrinkled seed shape. The genotype of the parent can then be written as capital R, capital Y, and smaller, capital small y. The cross between the two plant can be written down capital Y, capital R, and small y unite on fertilization to produce the F1 hybrid capital R, capital Y, small y. When Mendel self-hybridized the F1 plant, he found that 3/4 of the F2 plant had yellow seed and 1/4 had green. The yellow and green colored segregate in a ratio 3:1 between round and wrinkled seed. Shape also segregate in a 3:1 ratio, just like in a monohybrid cross. Figure 4.7. Result of a dihybrid cross where the two parents differed in two pairs of contrasting traits, seed color and seed shape. 4.3.1. Law of independent assortment. In the dihybrid cross, figure 4.7, the phenotype round yellow, wrinkled yellow, round green, and wrinkled green appeared in the ratio 9:3:3:1. Such a ratio was observed for several pair of character that Mendel studied. The ratio of 9:3:3:1 can be derived as a combination series of 3 yellow, 1 green with 3 round, 1 wrinkled. This derivation can be written as follows: 3 round, 1 wrinkled, 3 yellow, 1 green is equals to 9 round yellow, ratio 3 wrinkled yellow, ratio 3 round green, ratio 1 wrinkled green. Based upon such observation on dihybrid crosses between plants differing in two traits, Mendel proposed a second set of generalization that we call Mendel's law of independent assortment. The law states that when two pairs of traits are combined in dihybrid, segregation of one pair of character is independent of the other pair of character. The Punnett square can be effectively used to understand the independent segregation of the two pairs of genes during meiosis and the production of eggs and the pollen in the F1 capital R small r capital Y small y plant. Consider the segregation of one pair of gene small r and capital R. 50% of the gametes have the gene small r capital R and the other 50 have the small r. Now beside each gamete having either capital R or small r, if should also have the alleles capital Y or small y. The important thing to remember here is the segregation of 50% of the capital R and 50% small r is independent from the segregation of 50% of small y and 50% capital Y. Therefore 50% of the small r bearing gametes and has capital Y and the other 50% has small y. Similarly 50% of the capital R bearing gametes has capital Y and the other 50% has small y. Thus there is 4 genotype gametes, 4 types of pollen and 4 types of eggs. The 4 egg types of capital R small r, capital R small r capital Y and small r small y, each with a frequency of 25% or 1/4 of the total gamete produced. When you write down the 4 types of eggs or pollen on the two sides of a Punnett square, it is very easy to derive the composition of the zygote that give rise to the F2 plant. Figure 4.7. Although there are 16 squares, how many different types of genotypes and phenotypes are formed? Note them down in the format given. Can you, using the Punnett square data, works out of genotypic ratio at the F2 stage and fill in the format given? Is the genotypic ratio also 9:3:3:1? 4.3.2 Chromosomal theory of inheritance. Mendel published his work on inheritance of character in 1865, but for several reasons it remained unrecognized till 1940. Communication was not easy as it is now. These days his work could not be widely published. Secondly, his concept of genes infected in Mendel's work as stable as discrete units that control the expression of traits, and the pair of alleles which did not blend with each other was not accepted by the contemporary as an explanation for the apparently continuous variation seen in nature. Thirdly, Mendel's approach of using mathematics to explain biological phenomena was totally new and unacceptable to many of the biologists of his time. Finally, through Mendel's work suggested that factors were discrete units, he could not provide any physical proof for the existence of the factor or say what they were made of. In 1903, scientists D. Vries, Correns and von Tesselmark independently rediscovered Mendel's result on the inheritance of character. Also by the time, due to advancement in microscopy that were taking place, scientists were able to carefully observe cell division. This led to the discovery of structure in the nucleus that appeared in the double and divide just before each cell. These were called chromosome colored bodies, they were visualized by Stein. In 1902, the chromosome movement during meiosis had been worked out. Walter Sutton and Theodore Boveri noted that the behavior of the chromosome were parallel to the behavior of the genes. Are used chromosomal movement. Figure 4.8 to explain Mendel's law. Figure 4.3. Recall that you have studied the behavior of chromosome during meiosis equational division and during meiosis reduction division. The important thing to remember are that chromosome as well as genes occur in pair. The two alleles of a gene pair are located on homologous sides of the homologous chromosome. Table 4.3. A comparison between the behavior of chromosome and gene. During anaphase of meiosis 1, the two chromosome pair can align at the metaphase plate independently of each other. Figure 4.9. To understand this, compare the chromosome of 4 different colors on the left and right column. In the left column, possibility 1, orange and the green is segregating together, but in the right hand column, possibility 2, the orange chromosome is segregating with the red chromosome. Figure 4.9. Independent assortment of chromosomes. Sutton and Boveri argued that the pairing of the segregation of a pair of chromosomes would lead to the segregation of a pair of a factor that carried. Sutton united the knowledge of chromosomal segregation with Mendelian principle and called it the chromosomal theory of inheritance. Following this synthesis of ideas, experimental verification of the chromosomal theory of inheritance by Thomas Hunt Morgan and his colleagues led to discovering the basis for the variation that sexual reproduction produced. Morgan worked with the tiny fruit fly Drosophila melanogaster figure 4.10, which were found very suitable for such study. They could be grown on simple synthetic medium in the laboratory. They complete their life cycle in about 2 weeks and in single mating could produce a large number of progeny flies. Also, there was a clear differentiation of the sexes. The male and female flies are easily distinguishable. Also, he has many types of hereditary variation that can be seen with low power microscope. 4.3.3 Linkage and recombination. Morgan carried out several dihybrid crosses in Drosophila to study genes that were sex linked. The crosses were similar to the dihybrid cross carried out by the Mendel in pea. For example, Morgan hybridization yellow-bodied, white-eyed female to brown-bodied, red-eyed males in intercrossed their F1 progeny. He observed that the two genes did not segregate independently of each other, and the F2 ratio divided very significantly from the 9:3:3:1 ratio expected when the two genes are independent. Morgan and his group knew that the genes were located on the X chromosome section 4.4 and saw quickly that when the two genes in a dihybrid cross were situated on the same chromosome, the proportion of parental gene combination was much higher than the non-parental type. Morgan attributed this due to the physical association or linkage of the two genes and coined the term linkage to describe this physical association of genes on a chromosome, and the term recombination to describe the generation of non-parental gene combination. Figure 4.11 on the same chromosome, some genes were slightly linked, showed very low recombination. Figure 4.11 cross A, while other were loosely linked, showed higher recombination. Figure 4.11 cross B. For example, he found that genes white and yellow were very tightly linked and showed only 1.3% recombination, while white and miniature wing showed 37.2% recombination. His student Alfred Sturvant used the frequency of the recombination between the gene pairs on the same chromosome as a measure of the distance between genes and mapped their position on the chromosome. Today, genetic methods are extensively used as starting point in the sequencing of whole genome, as was done in the case of the Human Genome Sequencing Project, described later. 4.1 Linkage. Result of two hybrid cross conducted by Morgan. Cross A shows crossing between gene A and small w. Crossing B shows crossing between genes W and M. Here, dominant wild type allele are represented by the plus single sign in superscript. Note the strength of the linkage between small y and W is higher than W and M. 4.4 Polygenic inheritance. Mendel studied mainly described those traits that have distinct alternate forms, such as flower color, which are either purple or white. But if you look around, you will find that there are many traits which are not so distinct in their occurrence and are spread across a gradient. For example, in humans, we don't have just tall or short people as two distinct alternatives, but a whole range of possible heights. Such traits are generally controlled by 3 or more genes and thus called as polygenic trait. Besides the involvement of multiple genes, polygenic inheritance also takes into account the influence of the environment. Human skin color is another classic example for this. In a polygenic trait, the genotype reflects the contribution of each allele. The effect of each allele is additive. To understand this better, let us assume that 3 genes A, B, C control skin color in humans, with the dominant form A, B, C responsible for dark skin color and the recessive form small a, small b, small c for light skin color. The genotype with all the dominant allele capital A, capital A, capital B, capital B, capital C will have the darkest skin color that all with all the recessive alleles. Small a, small a, small b, small b, small c will have the lightest skin color as expected. The genotype with the 3 dominant allele and 3 recessive alleles will have an intermediate skin color in the manner the number of each type of allele in the genotype would determine the darkness or lightness of the skin in an individual. 4.5 Pleiotropy. We have so far seen the effect of a gene on a single phenotype or trait. There are, however, instances where a single gene can exhibit multiple phenotype expressions. Such gene is called pleiotropic gene. The underlying mechanism of pleiotropy in most cases is the effect of the gene on metabolic pathway which contribute toward the different phenotype. An example of this is the disease phenylketonuria, which occur in humans. This disease is caused by mutation in the gene that codes for the enzyme phenylalanine hydroxylase. Single gene mutation. This manifests itself through phenotypic expression characterized by mental retardation and a reduction in hair and skin pigment. 4.6 Sex determination. The mechanism of sex determination has always been puzzled for the genetics. The initial clue about the genetic chromosomal mechanism of sex determination can be traced back to some of the experiments carried out in insects. In fact, cytological observation made in the number of insects lead to the development of the concept of genetic chromosomal basis of sex determination. Hanking 1891 could trace a specific nuclear structure all through spermatogenesis in a few insects that it was also observed by them in that 50% of the sperm received this structure observed by him that 50% of the sperm received this structure after spermatogenesis, where the 50% of the sperm did not receive it. Hanking gave a name to this structure as the X body, but he could not explain its significance. Further investigating by other scientists lead to the conclusion that the X body of the Hanking was in fact a chromosome, and that is why it was given the name X chromosome. It was also observed that a large number of insects, the mechanism of sex determination is the XO-type. All eggs bear an additional X chromosome beside the other chromosome, autosome. On the other hand, some of the sperms bear the X chromosome, whereas some do not. Egg fertilized by sperm having an X chromosome become female, and those fertilized by sperm that do not have an X chromosome become males. Do you think the number of chromosomes in the male and female are equal due to the involvement of the X chromosome in the determination of the sex? It was designed to be the sex chromosome, and the rest of the chromosome were named as autosome. Grasshopper is an example of exotype of sex determination in which the males have only one X chromosomes beside the autosomes, whereas females have a pair of X chromosomes. These observations lead to the investigation of a number of species and understand the mechanism of sex determination in a number of other insects and mammals include man. XY type of sex determination is seen where both male and female have same number of chromosomes among the males, and X chromosome is present but its counterpart is distinctly smaller and called the Y chromosome. Female, however, have a pair of X chromosomes. Both males and females bear same number of autosomes. Hence, the male have autosome plus XY while female have autosome plus XX. In human beings, as in Drosophila, the males have one X and one Y chromosome, whereas female have a pair of X chromosome beside autosome. Figure 4.12ab. In the above descriptive, you have studied about two types of sex determining mechanism, exotype and XY type, but in both cases, males produce two different types of gametes, either with or without X chromosome, but some gametes with X chromosome and some with Y chromosome. Such type of sex determination mechanism is designed to be example of male heterogamy. In some other organism, example birds, a different mechanism of sex determination is observed. Figure 4.12c. In this case, the total number of chromosomes is same as in the both male and female, but two different types of gametes in terms of the sex. Chromosomes are produced by females. Female heterogamity. In order to have a distinction with the mechanism of sex determination described earlier, the two different sex chromosomes of a female bird has been designated to the Z and W chromosome. In these organisms, the females have one capital Z and one small capital W chromosome, whereas males have a pair of Z chromosome beside the autosome. 4.6.1 Sex determination in humans. It has already been mentioned that the sex determining mechanism in case of human is XY type. Out of 23 pair of chromosome present, 22 pairs are exactly same in both male and female. These are the autosomes. A pair of X chromosome are present in females, where the presence of X and Y chromosome are determined of the male characteristics. During spermatogenesis among males, two types of gametes are produced. 50% of the total sperm produced carry the X chromosome and the 50% Y chromosome. Beside the autosomes, female, however, produce only one type of ovum and an X chromosome. There is an equal probability of fertilization of the ovum with the sperm carrying either X and Y chromosome. In case of the ovum fertilized with the sperm carrying X chromosome, the zygote development into female XX and the fertilization of ovum with Y chromosome carrying sperm result into the female offspring. Thus, it is evident that if is the genetic makeup of the sperm that determines the sex of the child. It also evident that in each pregnancy there is always 50% probability of either male or female child. It is unfortunate that our society women are blamed for giving birth to female children and have been ostracized and ill-treated because of his false notation. 4.6.2 Sex determination in honeybee. The sex determination in honeybee is based upon the number of sets of chromosomes an individual receives. An offspring formed from the union of a sperm or an egg developed as a female queen or worker. As an unfertilized egg developed as a male drawn by means of parthenogenesis. This means that the males have half the number of chromosomes than that of the female. The females are diploid having 32 chromosomes. Males are haploid having 16 chromosomes. This is called as haplodiploid sex determination system and has special character features such as male produce sperm by mitosis. Figure 4.13. They do not have father and thus cannot have sons but have a grandfather and can have grandson. How is the sex determination mechanism different in the bird? Is the sperm or the egg responsible for the sex of the child? 4.7 Mutation. Mutation is a phenomenon which results in alternation of DNA sequence and consequently result in changes in the genotype and the phenotype of an organism. In addition to recombination, mutation is another phenomenon that leads to variation in DNA. As you will learn in chapter 5, one DNA helix runs continuously from one end of the to the other in each chromatid in a highly supercoiled form. Therefore, loss, deletion or gain, insertion, duplication of a segment of DNA result in alternation in chromosomes. Since genes are known to be located on chromosome, alternation in chromosome result in the abnormalities or abbreviations. Chromosomal abbreviations are commonly observed in cancer cells. In addition to the above, mutation also arises due to the change in a single base pair of DNA. This is known as point mutation. A classical example of such a mutation, sickle cell anemia. Deletion and insertion of base pair of DNA cause frame shift mutation. See chapter 5. The mechanism of mutation is beyond the scope of this discussion at this level. However, there are many chemical and physical characters that induce mutation. These are inferred as a mutagen, UV radiation that can cause mutations in an organism. It is mutagens. 4.8 Genetic disorder. 4.8.1 Pedigree analysis. The idea that the disorder are inherited has been prevailed in the human society since long. This is based on the inheritability of certain character feature on families. After the rediscovery of Mendel's work, the practice of analyzing inheritance pattern of the work, the practice of analyzing inheritance pattern of trait in human beings began, since it is evident that controlled crosses can be performed in pea plant and some other organism and not possible in case of human beings. A study of the family history about inheritance of a particular trait provide an alternative. Such an analysis of trait in several of generation of a family is called the pedigree analysis. In the pedigree analysis, the inheritance of a particular trait is present in the family tree over generations. In human genetic pedigree study provides a strong tool which is utilized to trace the inheritance of a specific trait, abnormality or disease. Some of the important standard symbols used in the pedigree analysis have been shown in figure 4.13. As you have studied in this chapter, each and every feature in an organism is controlled by one or the other gene located on the gene representative chromosome. DNA is the carrier of the genetic material. It is hence transmitted from one generation to other without any change or alternation. However, changes or alteration do take place occasionally. Such an alternation or changes in the genetic material is referred to as mutation. A number of disorders in human beings have been found to be associated with the inheritance of change or altered gene or chromosome. 4.8.2 Mendelian disorder. Broadly, genetic disorder may be grouped into two categories: Mendelian disorder and chromosomal disorder. Mendelian disorder are mainly determined by alternation or mutation in the single gene. These disorders are transmitted to the offspring on the same line as we have studied in the principle of inheritance. The pattern of inheritance of such Mendelian disorder can be traced in a family by the pedigree analysis. Most common and in eye prevalent Mendelian disorder are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia, etc. are the most important to mention here. That such Mendelian disorder may be dominant or recessive. By pedigree analysis, one can easily understand whether the trait in question is dominant or recessive. Similarly, the trait may also be linked to the X chromosome. As in the case of hemophilia, it is evident that the X chromosome recessive trait show transmission from character female to male progeny. A representative pedigree is shown in figure 4.14, pedigree for characters linked to both autosome and sex chromosomes. Blindness. It is the sex-linked recessive trait due to the defect in either red or green cone of eye, resulting in failure to discriminate between red or green color. This defect is due to mutation in certain genes present in the X chromosome. It occurs in about 88% of the males and only about 0.4% of females. This is because the genes that lead to red-green color blindness are on the X chromosome. Males have only one X chromosome and females have two. The son of a woman who carries the gene has a 50% of a chance of being color blind. The mother is not herself color blind because the gene is recessive. That means that its effect is suppressed by her matching dominant normal gene. A daughter will not normally be color blind unless her mother is carrier and her father is color blind. Hemophilia, the sex-linked recessive disease which shows its transmission from an affected carrier female to some of the male progeny, has been widely studied. In the disease, a single protein that is a part of the cascade of protein involved in the clotting of blood is affected. Due to this, in an infected individual, a simple cut will result to non-stop bleeding. This heterozygous female carrier for hemophilia may transmit this disease to son. The possibility of a female becoming a hemophilic is extremely rare, but because mother of such female has to be least carrier and the father should be hemophilic unviable. In the later stage of life, the family pedigree Queen Victoria shows a number of hemophilic descendants and she was a carrier of the disease. Sickle cell anemia. This is an autosomal linked recessive trait that can be transmitted from parents to the offspring when both the partners are carrier for the gene or heterozygous. The disease controlled by the single pair of allele HPA and HPS. Out of the three possible genotypes of homozygous individual for HPS show the disease phenotypic heterozygous HPA-HPS individual appears apparently unaffected, but they are carrier of the disease as there is 50% probability of transmission of mutant gene to the progeny, thus exhibiting sickle cell trait. Figure 4.15. The defect is caused by the substitution of glutamic acid by valine at the sixth position of the beta globin chain of the hemoglobin molecule. The substitution of amino acid in the globin protein result due to single base substitution at the sixth codon of the beta globin gene from GAG to GUG. The mutant hemoglobin molecule undergo polymerization under low oxygen tension, causing the change in the shape of the RBC from biconcave disc to elongate sickle cell structure. Figure 4.15. Phenylketonuria. This inborn error of metabolism is also inherited as that autosomal recessive trait affect individual lacks an enzyme that convert the amino acid phenylalanine into tyrosine. As a result of this, the phenylalanine is accumulated and converted into phenylpyruvic acid and other derivative. Accumulation of these in brain result in mental retardation. These are also excreted through urine because of its poor absorption by kidney. Thalassemia. This is also an autosomal linked recessive blood disease transmitted from parents to the offspring when both the partners are unaffected carrier for the gene or heterozygous. This defect could be due to either mutation or deletion, which ultimately result in the reduced rate of the synthesis of one of the globin chain, alpha and beta chain that make up hemoglobin. This caused the formation of abnormal hemoglobin molecule resulting into Anemia, which is characteristic of the disease thalassemia, can be classified according to which chain of the hemoglobin molecule is affected. In alpha thalassemia, production of globin chain is affected, while in beta thalassemia, production of beta globin chain is affected. Alpha thalassemia is controlled by two closely linked genes, HPA1 and HPA2, on chromosome 16 of each parent, and is observed due to mutation or deletion of one or more characters of the four genes. The more genes affected, the less alpha globin molecule produced, while beta thalassemia is controlled by a single gene, HBB, on chromosome 11 of each parent and occurs due to mutation of one or both the genes. Thalassemia differs from sickle cell anemia in that the former is quantitative problem of synthesizing a few globin molecules, while the later is a qualitative problem of synthesizing an incorrectly functioning globin. 4.8.3 Chromosomal disorder. The chromosomal disorder, on the other hand, are caused due to the absence or excess or abnormal arrangement of one or more chromosomes. Failure of segregation of chromatin during cell division result in the gain or loss of chromosome called aneuploidy. For example, Down syndrome result in the gain of extra copy of chromosome 21. Similarly, Turner syndrome results due to loss of an X chromosome in human females. Failure of cytokinesis after telophase stage of cell division result in an increase in a whole set of chromosomes in an organism, and this phenomenon is known as polyploidy. This condition is often seen in plant. The total number of chromosomes in a normal human cell is 4623 pair. Out of these, 22 are autosomes and one pair of chromosome and sex chromosome. Sometimes, though rarely, either an additional copy of a chromosome may be included in an individual may lack one or of any one pair of chromosome. These situations are known as trisomy or monosomy of a chromosome, respectively. Such a situation lead to a serious consequences in the individual. Down syndrome, Turner syndrome, Klinefelter syndrome are common example of chromosomal disorder. Down syndrome, the cause of this genetic disorder is the presence of an additional copy of the chromosome number 21, trisomy of 21. The disorder was first described by Langdon Down, 1866. The affected individual is short, saturated with small round head, furrowed tongue, and partially open mouth. Figure 4.16. Palm is broad with the characteristic palm crease. Physical, psychomotor, and mental development is retarded. Klinefelter syndrome, the genetic disorder, is also caused due to the presence of an additional copy of X chromosome result into the karyotype of 47XXY. Such an individual has overall masculine development. However, the feminine development of breast gynecomastia is also expressed. Figure 4.17a. Such individuals are sterile. Turner syndrome, such a disorder, is caused by the absence of one of the X chromosome 45 with XO. Such females are sterile as ovaries are rudimentary beside other features including lack of other secondary sexual characters. Figure 4.17b. Summary. Genetics is a branch of biology which deals with the principle of inheritance and its practice. Progeny resemble the parents in morphological and physiological feature has attracted the attention of many biologists. Mendel was the first to study the phenomena systematically. While studying the pattern of inheritance in pea plant of contrasting traits, Mendel proposed the law of inheritance, which are today referred to as Mendel's law of inheritance. He proposed that the factor, later named as genes regulating the character, are formed in pairs known as alleles. He observed that the expression of the characters in the offspring follow the definite pattern in different first generation F1 and second F2 and so on. Some characters are dominant over other. The dominant character are expressed when factors are in heterozygous condition. Law of dominance. The recessive traits are only expressed in homozygous condition. The characters never blend in heterozygous condition. A recessive character that was not expressed in heterozygous condition may be expressed again when it becomes homozygous. Hence, characters segregate while formation of gametes. Law of segregation. Not all characters show true dominance. Some characters show incomplete and some show codominance. When Mendel studied the inheritance of two characters together, it was found that a factor independently assort and combine in all permutation and combination. Law of independent assortment. Different combination of characters are theoretically represented in a square tabular form known as Punnett square. The factor now known as gene on chromosome regulating the character called the genotype, and the physical expression of the characters is called phenotype. After knowing that the genes are located on the chromosome, a good correlation was drawn between Mendel's law of segregation and assortment of chromosome during meiosis. The Mendel's law of were extended in the form of chromosomal theory of inheritance. Later, it was found that Mendel law of independent assortment does not hold true for the genes that were located on the same chromosome. These genes were called as linked genes. Closely located genes assorted together and distantly located genes due to recombination, assortment, development, linkage maps therefore correspond to arrangement of genes on a chromosome. Many genes were linked to sex also and called as sex-linked genes. The two sexes, males and female, were found to have a set of chromosomes which were common, and another set of chromosomes different. The chromosomes which were different in two sexes of named as sex chromosome. The remaining set of the two chromosomes as autosomes. In human, a normal female has 22 pair of autosomes and a pair of sex chromosome XX. A female has 22 pair of autosomes. A male has 22 pair of autosomes and a pair of sex chromosome XY. In chicken, sex chromosomes in male is RZZ and female RZW. Mutation defined as change in the genetic material. A point mutation is a change of a single base pair in DNA. Sickle cell anemia is caused due to the change of one base pair in the gene coding for beta chain globin. Inheritable mutation can be studied by generating a pedigree of a family. Some mutations have involved changes in whole set of chromosomes. Polyploidy or change in a subset of chromosome number, aneuploidy. This helps in understanding the mutation basis of the genetic disorder. Down syndrome is due to trisomy of chromosome 21, where there is an extra copy of chromosome 21 and subsequently the total number of chromosome becomes 47. Turner syndrome, one X chromosome missing and sex chromosome is SXO, and in Klinefelter syndrome the condition is XXY. These can be easily studied by analysis of karyotypes.